Today we are going to learn about transcription of DNA in prokaryotes. I'm going to be using a book as an analogy because whenever I think of transcription, I think of reading. We are now at a part of the DNA that I would describe as a gene. In transcription, RNA polymerase reads genes and adds bases to create RNA strands based off of those genes. Right here, we are at a part of the DNA called the promoter region. These two boxes, the -35 and the -10 box, or this is the ta-ta box, they basically tell RNA polymerase, hey, it's time to start recording the information, and it is time to start transcription. In order for RNA polymerase to start transcription, it needs two parts, a core enzyme and a sigma factor. Both of those together make a hollow enzyme. The sigma factor is the part that actually picks up on the -35 and -10 base sequences and understands that RNA polymerase needs to start adding RNTPs. We have reached the transcription site, which is denoted as the +1 area. This is where RNA polymerase starts transcription. And here's our DNA. Now, since we read from left to right, this example is going to have transcription going from left to right as if we are reading the DNA. Since we know that RNA polymerase adds RNTPs on the 3' side of the RNA, that means the 3' has to be towards the right, since we're going in the right direction. In order for that to occur, this means that the bottom strand will be our template strand, and then the top strand will just be our coding strand. I like to think of the coding strand in this terms as reading in between the lines of the book. After about 9 to 12 of these RNTPs down here are added to the RNA chain, the sigma factor drops off as it is no longer needed for the last two steps. As the RNA polymerase adds RNTPs to the RNA sequence, it also creates phosphodiester bonds to keep all these nucleotides together and to keep them in one chain. The step of chain elongation is just making the RNA chain longer with more RNTP bases. So in order to do that, the RNA polymerase continues to read the DNA until it terminates. I equate this to reading an entire chapter before finally finishing it. Now the last step is chain termination, which is just the cutting off of the RNA chain and making it one full strand of RNA for translation. There are two ways an RNA chain can terminate, and both of them use this sequence of adenines in the template strand to make a bunch of uracils. The first way is using what's called an inverted repeat. As you can see, if you go backwards from these two sections here, you can see that they match up. And so since they match up, they want to bond with each other. Since these inverted groups want to bond with each other, they are going to break from the DNA and they are going to fold back so that the inverted repeated groups line up like this, as you can see. And this creates a termination hairpin. Now, let's say we come across a gene that wants to make sugar, as denoted in the candy picture right here. And we have our promoter region, but we don't have our -10 box. It's not there in the gene. Well, in order for transcription to occur, both of those boxes need to be present. So if that gene wasn't there, then the RNA polymerase and hollow enzyme group would not be able to transcribe the gene, which means that the gene for making sugars would not work and the bacteria would most likely die.